Alright, the rest of the RNA story. So, mRNA, check. Now it's time for us to get into the others. Our next one is tRNA. tRNA, transfer RNA. That's what it's called, and here's what it looks like. Make sure that you draw a short end here. This is 3 prime, right there. Obviously, that means that this is 5 prime. This part, let's call this the 3 prime acceptor end. Down here, we're going to identify 3 parts. We'll point these out later. This whole thing here is called the anti-codon loop. First time, surprisingly, that you've heard codon today. We haven't mentioned it. We'll mention it soon. So we have codon, anti-codon. This is an anti-codon loop. What we will do is follow the instructions, follow what's guided for, what's outlined in your book to come up with a cartoon. And here's what our cartoon is going to be. So there we have our anti-codon loop is very, very exaggerated. Boom, boom, boom. Units of 3. And of course these are going to be 3 letters in our language. And our language in RNA is adenine, guanine, cytosine, and uracil, if you remember. Then up here, what we got going up here at the very top is our acceptor end. And this could be 1. And what we're going to do is take this cartoon of a transfer RNA, and we're going to say plus the specific amino acid for it, and that's a specific relationship, equal our next bit, and that bit is going to be let's stick our amino acid here. So this is our amino acid specific to this to this 3-letter code. We'll talk about this later. This guy is called charged. Charged. So we have a tRNA in a state of not having an amino acid. That's this guy. And then we have one that has an amino acid. So these things are kind of like little carts in the factory, and they pick up a specific amino acid to the cart, and they bring it to the site, which is the ribosome. That's our next part, is the ribosome, and I'll see you pretty soon for that. Okay, so now it's time for us to take a look at ribosomal RNA. But before we go too far, just remember that the tRNA and the rRNA were under the control, similarly, of RNA polymerases. We outlined those at the very beginning. So remember, there's 3 types of RNA polymerases because there's 3 types of RNA. Okay, so here we go for ribosomal RNA. So there we have it. We're going to take a look at this. There's 2 subunits. They make ribosomes. And we'll see they have 2 subunits in a second. I jumped the gun. We actually will see these ribosomes on rough ER. We'll also see these in prokaryotes. They're different. We'll also see in mitochondria, and we'll also see in chloroplasts and actually other things. But that's it for us. These ones here will be different from these ones. So we're obviously going to be focusing on the eukaryotic ones. So 2 subunits. And these subunits are actually, kind of interestingly, there's this old system called sedimentation where you take large, dense molecules and where they occur in a kind of a gradient of density. And the 2 subunits of ribosomes came out as being large and small. And the large subunit was called the 60S. And the S stands for units of sedimentation called Svedberg units. Svedberg units is what they are, and this is a sedimentation phenomenon, so it's kind of old 20th century beautiful stuff. And then we have also the 40S, and when you add these together, the full ribosome equals an 80S in eukaryotes. So this is all kind of eukaryote ribosome business. And if we were actually to draw a very nice cartoon of a ribosome, here's what it would look like. So this kind of looks like a big kind of chubby catcher's mitt part. And then this part here, that's a fairly decent cartoon of this. Okay, so this is our, and then as you might guess, the 60S is this big part, and the 40S is that part. And what we're actually going to see in this ribosome are these sites. So we have these 3 sites, and we're going to go ahead and name these. This is E, this is P, this is A. It'll be the next board before we get into the details of this, but this is EPA, and that's what the ribosome looks like. So, matter of fact, let's take a look at these EPA. So it has an E site. I got enough on this board. So the E site is the site that we call actually the exit site. And the exit site is, well, let's just name them. The next one there is the P site. And the P site is called the peptidal site. That should ring a bell. Remember polypeptide, remember proteins, chains of proteins, polypeptides. So the peptidal site. And then we have the A site, which is our last site, which is called, for lack of a better term, it's called the aminoacyl site. But I like to take advantage of the letter, and for me it means arrival. So that's those are our sites, and these are our ribosomes. So now this is just a very cartoonistic view, because remember that this is made out of single-stranded RNA that's looped and twisted and turned on itself to make a three-dimensional reality, because ribosomes truly are three-dimensional. But they're made out of nucleic acids, so they're not proteins. It's cool. Alright, so we got that going for us. Let's take a peek at how this thing works. And then we can actually define the process of translation. So that's that. Uh-oh, just lost a beautiful big piece of chalk. This has been a very expensive day in the chalk department. Okay. Alright, here we go. So we've got our picture of that. Let's draw it up here. So here's our catcher's mitt. here's this business. And so here's our large subunit, our small subunit. Here's our E, here's our P, and here's our A. Maybe not putting those, yeah, that'll work. And then what I wanted to do was to show you that here's what's going to go on here. Going this way, 5' end, 3' end, M, R, N, A, all the stuff that we talked about before, spliced, beautiful, and it's cruising through this site. And then, of course, what we want to do is identify that there is going to be base, 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 units of base, 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 base. This is all a language of 3 letters. And then, complementary to that, we're going to have base, 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 base. Now, no matter what you make out of this, you would know that if we were just focused right here on this one, if that was an A, this of course is RNA, that's a U. If that next one over is a C, that's a G. So remember our complementary base pairing in all of this process. So CG, AU, and that's RNA lingo. And so what we're going to therefore have in blue here would be the glorious language. These, that bit of 3, that bit of 3, and that bit of 3 are codons. And those codons are going to be 3 base sequences that code for a specific amino acid. And then these here, of course, those units of 3, anti-codons. Wow. We are starting to look really, really good. We need to get a little more detail here. Let's suppose that we took one of these and looked at a phenomenon. Let's look at this process up close and say that, okay, here we are. So I've done a big blow up here, and let's say that we have this beautiful bit of, and of course the whole thing is kind of this way and then this way. The whole thing is lined up here, and we're going to say that, okay, here's a C, here's a G, here's an A, here's a G, here's an A, here's an A, here's a and a U and a G. So on the complementary to that, G, C, U, C, U, U, and a G, oops, go with U, and then A, and then C. So we have that. Now, what we want to do is show this bit right here that if this part was actually occupied with transfer RNA, and we have one that comes in, this guy comes in, A for arrival. This guy's going to come in charged, so we actually have to look at a table. I'm going to deal with this a lot more. I'm going to show you this table. So what I'm going to do right now is I'm going to go to our table so you can actually see what these things are. We're going to look at these bases on messenger RNA in the table, first base, second base, third base, and we'll see that on PowerPoint. And each one of these is going to give us the identity. Now, this guy here, although it's not here, was arginine. This guy actually is here, and this one is my favorite, so I'm going to stick it up here. This is This is GLU. That's glutamate, glutamic acid. Monosodium glutamate. You remember my fun thing? The salt version of that amino acid? Well, that's that. And then we'll assume that something else was stuck there. Here's what's coming in. What's coming in is AUG. Let me get AUG. AUG. Oh, I can't have that. My goodness. I'm going to have to change this, everybody, because that was a start codon. Let's make that a C. CUG. So a CUG is going to be leucine. So leucine I'm going to draw in green. And that is, leucine. That is leucine. Let's just double check that. Make sure that's LEU. Why can't I remember how to spell leucine? LEU. Leucine, glutamate, arginine. These are all names of amino acids. You can go way back to the amino acid table, well back in the semester, and figure that out. But what we want to also identify is that this thing has, if this has gone on for a while, which is implied by the length of messenger RNA, we would have a chain, you know. So there'd be an amino acid, here'd be an amino acid, all stuck together. A beautiful, glorious polypeptide. There's a leucine right there. We'll stick an arginine right here. We'll stick whatever this is. We'll stick whatever this is again. This is our polypeptide right here. Let's put some more on just so we can kind of see the glory, see the beauty of this thing. Oh, let's do a purple one. Okay, so this whole thing, this whole kind of sequence of beautiful amino acids, this whole business is our polypeptide. I'm going to actually erase this so we can see it better. And let's erase the mothership over here, because now we're looking at a bigger picture. I'm going to kind of isolate what we're doing. So this whole thing here is basically our growing polypeptide. Now, because of this information, we need to identify what is happening at these places. There's an enzyme that's involved here that is doing some fantastic work. Let's see. So right here is under the control of an enzyme. Boy, I need something. I need some punch. Maybe I should just grab a little tiny piece of white chalk. So right here is a process. What's working, or actually I should change the arrowhead. What's working right here is an enzyme catalyzed dehydration synthesis. That is what's happening. And of course, we know that dehydration synthesis is how we attach amino acids together between the amine and the carboxyl group. We make water. So what's happening there, that enzyme is called, the enzyme in the action is called, let me get the specific name, peptidyl transferase. So there's an enzyme that catalyzes that, and it grows the polypeptide, and of course this is growth by dehydration synthesis. And of course the transfer part is it's being transferred from the leucine and stuck here, so that's going to happen there. Now you know that this is the exit site, so really this guy actually goes off into the cytoplasm. This guy goes off into the cytoplasm with its GCU, and this will go back back for more. So back for more for another, in this case, arginine. So this is in the cytoplasm. Back for another one is it leaves, and so that's for that's what we should say is exit. Okay, so now we've kind of put together the whole process of what's going on here. It's fabulous. We keep chinking through in units of 3. Remember, we call these codons, and then we call these things anticodons here. And etc., etc. So look at that. Spectacular. Now, the other thing that I wanted to do was to put this in perspective of where this occurs. The last thing we want to do is see where this whole, how this is arranged. God, that's a good board. I almost don't want to erase that board. It is so beautiful. But I'm going to have to, because we got more to do. And so let's do it. God. Damn. Good job. I guess I'll do it now. Okay, so here we go. Let's suppose that this right here is, so this is basically ER, you know, and we could look at it like this all day and night, and what we'll see is the big subunit, the small subunit. The big subunit, the small subunit. Whoa, I don't need that to go in. Come on now. Okay. There's a nice little pore that we need to kind of line up with. Let's get our drawing together here. I mean that to be, I keep drawing it too high. This is how these ribosomes are in relation to the endoplasmic reticulum. Okay, there we have it. And let's draw some sites. That's spectacular. So this is the ER. This is basically, therefore, this is kind of the lumen then of inside. This is the lumen. This is inside. So this is therefore cytoplasm. This is cytoplasm. Every one of these things is a ribosome. And then what we would see is actually each of these coming right off of the tops of their, I'll get it multicolored and spectacular. This is going to be a great board as well. And right off of the P, the P site, I'm going to actually set one little amino acid that's in place. And then, of course, we'll see ones that are being carried in. So these guys could be carried in. And set these up as little red ones. Oops. And let's see. Let's get some others here. This is just a lot of fun. Okay, I didn't even stick anything here yet. And this nice orange one. And then I'm just doing different colors to kind of show you that, you know, we're dealing with chains of amino acids that are working their way into the system, and they are now within the endomembrane system. If as they continue to be synthesized and carry in, they carry into the lumen of the endoplasmic reticulum. So these growing polypeptides are just growing and growing and getting beautiful. Of course, this is primary structure of protein. I'm just doing a little bit of crazy. Let's stick some together so you get the idea. We'll make this one be our longest one at the bottom. And look at that. Getting orange amino acid. Let's get another red. So look what's happening here. I'll make this, finish this because I've actually started some action here. So every one of these is an amino acid in a growing chain. You know, this one is small. And then the last part of this glorious drawing that we need to get together is we need to get together some so look at that, growing polypeptides. And guess what? They're in here. Did you remember what's going to happen? So this endoplasmic reticulum, they're inside membrane now. They're in the endomembrane system. Note, polypeptides are in the endomembrane system. They're in the system. And because of that, what happens? Next stop, next stop, next stop. How about some folding? Remember, we've got to get these polypeptides are primary protein structure. Folding. Let's get secondary, tertiary, quaternary if necessary. Somewhere in here could be some spectacular vesicle work where we extrude, we pinch off of the endoplasmic reticulum, travel to the Golgi. So look at that. That is extraordinary. And that's a beautiful board as well. At the very, very, very end of the process, it's the last thing we need to talk about. I hope you guys have a pretty good snap of this board. It's really nice. That's far, far superior to what I would draw if we were in the classroom on the whiteboard. This is good stuff. Look at that. It's great. So this is polypeptide chains, primary structure of proteins, in the perfect space that they need to be for all of that extraordinary twisting and turning. You know, secondary structures. What causes that? Do you remember? Alpha helices, beta pleated sheets. That would be basically hydrogen bonding from the, between the kind of the amine and carboxyl group of the chain. And then, of course, the tertiary structure folds in with beautiful, beautiful interrelationships between different kinds of amino acid R groups, such as some covalent bonding, like the disulfide linkage, some hydrogen bonding, van der Waals forces, ionic forces. Remember, glutamate's an acidic R group, so it actually dissociates, and then it's sitting there as a big fat negative charge, right, for the carboxyl group. Great, great stuff. I'm hoping that that little side discussion has brought you back to the kind of early mid of the semester, where you can kind of see that, hey man, we have come a long, long way. We put this all together. The whole story is here. Wow. Okay, so as promised, with that little spiel, I wanted to tell you that the last thing that we need to kind of address, still leaving the glory of this board, is that in the end, what is going to happen is that, at the end of the run, because, oh God, do you realize that I didn't put these messenger RNAs in here? Here's our messengers. Remember? Here's a big long one that's just getting started, Same thing there. All of these are mRNAs. It's a good thing that I did not get rid of that whole thing. And of course, you want to remember that it's 5 prime, 5 prime, 5 prime, 5 prime, 5 prime, 5 prime. Then, of course, 3 prime on the other end. That's how it travels. Whew, sorry about that. That's what those messenger RNAs that we sent there did. Okay, so at the whole end of the story, what is going to happen is that the ribosome, at the end of the mRNA, what we're going to see here is a stop codon. A stop, I should probably keep it in blue. A stop codon. So we have stop codons that are specifically in the language. We have several of them. UAA, UAG. Here they are. UAA, UAG, and UGA are universal stop codons. They don't carry an amino acid. They say, stop, we're done. And when that stop signal is reached, when you get that, what you get in the process is ribosome complex dissociation. Now, the other thing that I didn't do, so we've got stop here, we've got stop here somewhere, we've got stop, we've got stop, we've got stop, we got all these stops. At the very beginning, we have to have a start. And those may not be showing up, but let's take a look at those. I actually made the mistake earlier of putting the codon for start in my first A-site rendition of transfer RNA. And I had to change it. I hope you caught that. So this next part then is the other side of the story. So on the other end of the deal, what we have here is at the beginning, after our 5 cap, we actually have a start codon. And it's a universal. There's only one, so we call it universal. We call it universal, and it is a start codon. Start codon, and that start codon is AUG. So AUG, green light for start, UAA, UAG, UGA. So here we sit, process of translation. So this is in the cytoplasm. What we don't see here is the endoplasmic reticulum membrane, which would be like right here. And we actually see the polypeptide passing through it, however. So this is our growing polypeptide. In certain cases, they have identified because of the code, they've identified the players. So here's our codon, UGG, UUU, GGG. Those should all equate to things. I'm going to go to the next slide right now so you can see that. So let's actually take a look at UUU and then look at GGG on the genetic code. So first base, second base, third base. That's how this works. This is first mRNA base on the 5' end of the codon. Second and third. So we want to find U. And UUU is just right there. So that's PHE, phenylalanine. Let's go up and see if that was it. BHE is attached to this to this transfer RNA. So here we have it all. This tan stuff is ribosomal RNA. These beautiful light blue things, transfer RNA in the cartoonistic form. And then this red thing, in a less cartoonistic form, is our messenger RNA that had already been through all of that processing. So we had spliced and gotten rid of the introns and linked together the exons, and it's fabulous. So this thing's passing through. Remember, it's EPA. So E is this guy, and we have an exit phenomenon going on right now. That thing is going to go to the cytoplasm and get recharged with the amino acid that it is specific for. Here, you actually have charged transfer RNAs that are bringing their amino acids in. And in this case, it is this case it's a GGC. Let's take a look at GGC and make sure it matches. GGC is glycine, GLY. That's what that symbolizes. So that's an amino acid glycine. Go back up and see that the glycine is attached. And remember, that is charged. Charged. So this guy comes into the A site, the P site's in the middle. What they don't show here is that fabulous process of dehydration synthesis that will actually involve both a transfer, release basically, from the tRNA and attachment to, in this case, the phenylalanine. And this growing polypeptide will grow by 1, and so on and so forth, as units of 3. 3, 3, 3, 3, 3. This will go all the way until we have a stop codon. It actually started way up here on the 5' end somewhere with a start codon. So I need to go back to the genetic table and show you that. AUG is the only start codon that we have. It is the universal start codon. So when that thing, it's got its 5' cap, and that 5' cap is the guanine triphosphate, and then it chinks through after that, and when it hits there's a UTR, and then when it hits the AUG, it's time to start building. And then we go through, and at the very end of the process, it would stop when it gets the codon signal of either a UAA or a UAG or a UGA. So 3 options for the stop codon, and that signals time to dissociate. So ribosome and the messenger RNA break apart. An extraordinary process, constantly going in every one of our cells. Wow, wow, wow. Okay, so hopefully this puts a pretty good kind of structure to your understanding of from gene to protein. What we need to do is sit back, take a look at this extraordinary big picture, see that we have actually put the fundamental scaffold steps in. You know all of the big parts, you know all the big players, and then celebrate some of that super fine detail that we threw in as well. The whole idea is to kind of maybe make you want to learn more, sink your teeth into that. Some of you may actually go on and actually take a course in genetics. And if you take a course in genetics, you're going to be deeply mired into the details of this. And of course, if you really become addicted to it, then you're into classes in molecular biology and cellular biology, or maybe you do biochemistry. This is where that path leads. And it's exciting, constantly changing, extraordinarily important in the world today. I mean, none of our success in flattening the curve and doing everything that we're doing with the coronavirus, actually some of it would, because it's very, very old epidemiological techniques to do that, right? But our understanding of this coronavirus is so much more intensified by this whole molecular biology field, the science of molecular biology. And if you think just knowing the virus is important, to know the virus enables so much more. just put in coronavirus into a search engine and vaccine development. And you are going to see full throttle, faster than ever before, race by scientists all over the world. Scientists working in applied science, like for companies, trying to develop these things. We've got a couple of leaders right here in our country, as well as academics, like professors that are experts in vaccine development that are working. And sometimes they merge. Universities and industry come together. And they are working day and night, pressing as hard as they can. And what they're working on and the techniques that they're doing, molecular biology. One more thing about molecular biology. We need to test for this coronavirus epidemic. That's what everybody says. We've got different kinds of tests. Sometimes we're looking for proteins. Sometimes we're looking for, for these reactions between proteins and proteins and other kinds of molecules. We've got all sorts of spectacular biochemistry and molecular biology involved in the testing procedures that we have. Of course, the world is completely stressed for the flow of the materials that are necessary to do the kinds of work that you do in molecular biology. They're solving that. It's a little bit easier to make some chemical reagents than it is to make ventilators, if you've been following this whole deal. But these are exciting times, and I guess the point that I'm trying to make is just first off, give a broad kind of appreciation to the scientific community that has worked so hard to develop these techniques, and then the followers to learn these techniques, the innovators, because without them, man, we're nowhere. And then give yourself a lot of credit for hammering through this class and getting up on top of this mountain, because now you are equipped with this basic understanding that so many people on the world, on the planet, just don't have and never will. You have the gift of education, and you've worked to get this base of knowledge, and with that knowledge comes power, and with that knowledge comes responsibility. Tell the story. It's out there. Okay, so I think that's kind of me saying that's the end of our course as I know it. And it was a weird one. It was a beautiful one. I really enjoyed it. From the front end, getting to meet you and see you in person, to the back end of lecturing to the whiteboard, to the chalkboard, and then having strange virtual meetings with some of you where I saw you as initials on the screen. It still doesn't matter. I know who you are. I know what you've done. I know that it's stressful, but I know that at the bare minimum, you have been bettered by something that happened in this class. That's all I got, and I hope to see you continuing your education, and maybe even in my future classrooms, whether they be virtual or on campus. Ciao.